Thank you very much. I'll make this brief. I was excited when I saw the topic of epigenetics for this year's symposia. And when I looked at the speakers that were getting lined up, I was really hoping we would have some cancer-related topics. And so I was pleased when Joya accepted the invitation, in part because of her link with cancer epigenetics, but also our link with MD Anderson Cancer Center, where she comes from. Some of you may not realize, but we are the only academic affiliated partner in the MD Anderson Physicians Network. And that puts us in a pretty unique place, I think, to expand our research collaboration with MD Anderson and Ellis Fischel and the rest of the campus. And so in our discussions that we had yesterday, I think there are several areas where that might happen. Joya went to LSU, where she got her bachelor's. She then got her PhD at the University of Texas Health Sciences, and did 2 postdocs, one in Stockholm and one at the Mayo Clinic, and has been back at MD Anderson in a role in pediatrics as an associate professor. She's got a lot of interesting areas that I think some of you will find some similarities with your own work, but importantly, this ties into our previous talk in that she's going to discuss pediatric cancer. So if we're looking at things that are related to the entire lifespan, I think this will be an interesting concept of how pediatric cancers differ from adult cancers. So help me welcome Joya.
Thank you, Carolyn, for that kind introduction. I really have you to credit for insisting on a cancer research being here and someone from MD Anderson being here, and I've appreciated the opportunity to visit. Yesterday and the day before, I've been dazzled by the array of scientists that I've met that are working here, and I'm sure there'll be opportunities to collaborate in the future. I wanted to thank Mary and the committee for organizing such an interesting symposium. I've never given a talk in a symposium that included historians and philosophers, so this is really innovative and forward-thinking. And I also want to thank my host, Kristen Taylor, who's taken time to run me around, and I've gotten to learn about her exciting research too. So it's almost the end of the session, of the symposium, and I hope you're not feeling this way. If you are, I just have a couple of take-home messages that I want to tell you now, so that if you are feeling this way, I just want to impress upon you the excitement about epigenetics in the realm of therapeutics, not just for cancer, but for neurodegenerative diseases, for a number of human pathological conditions. And so that's an aspect that I think hasn't been touched upon as yet in this symposium, and I'll give you a sense of where the field is heading in terms of cancer therapeutics and kind of harnessing the power of epigenetics. So this issue has come up time and time again. I'll weigh in on my definition of epigenetics. It is heritable, but what we really focus on is whether or not gene expression has changed. And so in response to the question that was asked yesterday about RNAi being an epigenetic alteration, I would argue yes, because it does alter gene expression. And so it has to be independent of alterations in DNA sequence. So that's the definition of epigenetics that I ascribe to and kind of convey whenever I teach students about epigenetics or introduce the topic to new folks in the lab. So this is a review article from The Scientist, which I think if you're interested in getting an introduction to the field is really useful. It talks about the organization of DNA and the fact that there's all of these modifications that occur on both DNA and on histone proteins that ultimately influence gene expression. So at the end of the day, it's really all about packaging, which you heard a little bit about in the last talk. The term chromatin refers to the complex of DNA and histones, and you may hear the term chromatin biology used interchangeably with epigenetics, and it's really reflecting this kind of concept of packaging. So again, chromatin is just the complex of DNA and histone proteins. Nucleosomes is another term that you might hear in the field of epigenetics, and these are the functional units of chromatin, and they're composed of sets of these histone proteins that DNA is actually coiled around, and there's distinct DNA pieces that are coiled around these histone proteins. There are 147 base pairs, and then there's these kind of spaces in between the pieces that are coiled and connected to histones, and those are usually 20 to 50 base pairs of linker DNA in between these histone complexes. And so the central dogma of epigenetics is that unwrapped DNA is more accessible than wrapped DNA. So the tighter DNA is wound, the less accessible it'll be for gene transcription. The tighter it is, the less accessible it'll be. So it's really an issue of this wrapping versus unwrapping. So what regulates this wrapping versus unwrapping? You may have heard the term epigenetic marks. And so these marks occur both on DNA as well as on these histone proteins. And this is kind of showing you the interplay between the DNA marks and the histone marks. On DNA, the primary marks are methyl groups. On histone proteins, there's a huge array of marks, and that's where kind of my interest lies in terms of trying to understand the histone code, the significance of these marks on the tails of histone proteins, as May explained, that ultimately influence interactions with DNA and influence gene expression. So the concept of marks has further definitions because marks can be added, marks can be subtracted, and marks can be read. And so there's frequent referring to writers, erasers, and readers. Epigenetic writers are adding chemical modifications to DNA or histones. So this is a broad term, so it's inclusive of the methylation marks that are being added to DNA. It's inclusive of all the many marks that are added to histones, but it's basically adding a mark. If you're subtracting a mark, then the enzyme is classified as an epigenetic eraser. And there's demethylases for histones, there's deacetylases, depends on the mark, so this is a generic term. And also epigenetic readers are basically enabling recognition of another complex of proteins to DNA. So they're not adding a mark or subtracting a mark, they're enabling protein-protein interaction, so they're a recognition motif. So these are some terms that you might come across in terms of learning more about epigenetics and these packaging issues. So why are we interested in epigenetics in a cancer setting? Well, the basic idea is that the process of transformation of normal tissue to tumor tissue involves reprogramming, and that reprogramming is partly epigenetic in nature. And so there are examples of comparisons of DNA methylation patterns, of analysis of histone marks, where comparisons have been done with normal tissue and tumor tissue, and there's differences. And that's really reflected in data that has emerged from sequencing of many different types of cancer, where it's seen that there's alterations in epigenetic enzymes in numerous cancer types. And so the part B of this figure is only focusing on chromatin regulators and is identifying altered expression of epigenetic enzymes. So this is inclusive of readers, writers, and erasers that are tracking with different disease types, and these are all cancers. And you can see that it's not limited to any one specific organ. It's not limited to any single tissue. There's lots of different alterations in chromatin regulators in many different types of cancer. Even more kind of confusingly, these alterations are not consistently loss of function or gain of function. So in some instances, they are loss of function. In some instances, they're gain of function. So they really have to be evaluated on a case-by-case basis. And it's not clear that these altered chromatin regulators are by themselves oncogenic. So they may be setting the stage for an additional event, perhaps an environmental exposure, perhaps cooperativity with an existing oncogene, an existing genetic effect that's oncogenic. So this is quite correlative in terms of seeing a change, either up or down, and it's not even clear for some of these whether the change in expression really affects a change in function of the enzyme. So there's that element. There's the question of whether loss or gain of function is oncogenic by itself or with something else. But nonetheless, it's striking that there's so many mutations in epigenetic enzymes prevailing across so many cancer types. So pediatric cancer, this is, I think, a little bit misunderstood because people talk about pediatric cancer as if it's one disease, and of course it is not one disease. There's lots of advocacy about breast cancer, about prostate cancer, but when people talk about pediatric cancers, they're talking about a wide spectrum of diseases. And fortunately, there have been a lot of successes in treating pediatric leukemia, such that the cure rate is upwards of 85%. So there's a perception that many pediatric cancer patients, or most pediatric cancer patients, do pretty well. It doesn't need to be earmarked for grant funding, but the reality is that even for pediatric leukemia, where we have a good cure rate, we need to do better. There's relapsed and refractory patients that don't do as well. The late effects that are associated with pediatric leukemia treatment can really influence quality of life. The drugs that are being used have been used for decades, and we need to find more selective ways to treat pediatric cancers. So these are some materials from some of the fundraising entities, Alex's Lemonade Stand, as well as Cure Search, that try to raise awareness about pediatric cancer and really highlight the fact that it needs additional researchers and additional research dollars, and that it's a spectrum of diseases and it is far from cured. So in terms of pediatric cancers, this is a pie graph showing you the array of different types of pediatric cancers. You can quickly get a sense of what the most common pediatric cancers are. Leukemias are the majority of pediatric cancers. They're either acute myelogenous leukemia or acute lymphocytic leukemia. They're lumped together in this purple wedge. CNS tumors are the other major chunk of this pie in terms of pediatric cancers, and this is also not really simply one disease. There's different types of CNS tumors. I'll be talking today about glioblastoma, which is one of the worst pediatric cancers to get. In particular, the subtype of pediatric glioblastoma that I'll be speaking of has a less than 10% survival rate 2 years after diagnosis. So it has not been touched in terms of improvement by the existing treatments. So of course there's lots of other subcategories of pediatric cancers, and I'm not trying to minimize the importance or impact of these other subtypes. I know that there are people in this room who have had their lives touched by some of these other subtypes, but in the interest of focusing, I'm going to talk about leukemias and brain tumors today. So some of you may be aware of another institution in Missouri that's kind of down the road. I'm at the Children's Cancer Hospital in Houston, Texas, and so we're aware of another Children's Cancer Institute, and they've collaborated and done a lot of amazing work that they're making public in terms of sequencing pediatric genomes. And so this is a figure from one of the papers from the St. Jude's and WashU Pediatric Cancer Genome Project, where they've sequenced 1,000 pediatric cancer genomes, and there are a large number of these epigenetic enzymes showing up as mutated. And you can see that there are writers, there are proteins that bind the writers that therefore may regulate the activity of the writers. There are histone proteins themselves, which is really interesting and was a big surprise in terms of one of the major findings of this work. There are mutations in erasers, so the enzymes that are taking off the marks. mutations in proteins that bind the erasers, but this was a major theme in their sequencing findings, that these epigenetic enzymes are frequently mutated. Again, the significance of each of the mutation, I think, has to be carefully delineated, just like I explained for the adult cancers. Is the mutation activating and inactivating? Is it silent? What's the biological significance? Is it oncogenic on its own? Does it cooperate with other oncogenes? So that's kind of the grain of salt in kind of getting carried away with this data. So in the context of pediatric leukemias, there are definitely distinct epigenetic features that distinguish pediatric leukemias from adult leukemias. And this is a pie chart that's focusing on translocation of MLL. And MLL is the mixed lineage leukemia gene. It is a DNA binding protein that is methylating a specific residue, H3K4. So pop quiz, is this a reader, writer, or eraser? Anyone? Writer, very good. Yay, okay, so it's methylating a mark, right? So it's adding a mark, so it is a writer. Kristen, I think that you had an unfair advantage, but thank you. Okay, so it's a writer, and it actually acts as a translocation partner and fuses with other genes. So it kind of inappropriately breaks off and finds a partner, and this fusion gene appears. And the fusion gene has different rates of appearance in pediatric versus adult leukemias. And this can be dissected down to the two major types of acute leukemias, ALL and AML. And if you compare the pie graphs for ALL, for example, you'll see that the MLL AF9 fusion is much more common in pediatric ALL than in adult ALL. It's 18% of pediatric ALL cases, only 2% of adult ALL cases. So that's a putative target for pediatric ALL. Well, how can this alteration actually be targeted? Well, we have to understand how MLL-AF9 is working and how the entire complex works. And so this is where things get hairy, because MLL-AF9 has multiple proteins that it interacts with. The current strategy is to think about some of these binding partners and to work to inhibit them and ultimately inhibit the gene expression pattern that MLL-AF9 is driving. And this particular MLL fusion promotes the expression of development-related genes, specifically the Hox family of genes, which are thought to go awry and lead to kind of uncontrolled growth. This is a common theme in pediatric cancers, that development-related genes kind of get turned on and go crazy, in contrast to a lot of adult tumors where an environmental component is more at play. So some of the strategies that are being used to target MLL rearranged leukemias are to target this protein called DOT1L, which interacts with MLL-AF9, and then to target proteins that contain bromodomains that are kind of indirectly interacting with MLL-AF9. And there are representative medicines for each of these classes that are currently in early clinical trials. So that's an example of a kind of unique feature of pediatric leukemia where the biology is guiding where the trials are going. In pediatric glioblastoma, there's similar striking differences between adult and pediatric cases. And so this again arose from sequencing studies in which adult GBM glioblastoma cases were compared to pediatric glioblastoma cases. And there was a subset of pediatric glioblastoma called diffuse pontine glioma, which is really deadly. So this is the type of brain tumor where kids have a 10% survival rate 2 years after diagnosis. It's unique in that it arises in the brainstem, and what they found was that there were very specific mutations, single amino acid mutations in histone proteins and in histone variants, so histone H3.3 and H3.1, these were present in 78% of these pediatric brain tumor patients. That's a really high rate. And so people kind of like were shocked that this was the case, but it has, and it was only discovered like 2 years ago, so it's a pretty new finding. Even more interesting is the fact that now we know enough about brain tumors and mutations to kind of track the incidence of some of these mutations with age of incidence and with location of the tumor. So these two columns here are referring to the point mutations that are present in the pediatric cancers. The point mutations are a K27 point mutation and a G34 point mutation, again, in the histone proteins. And this is showing age distribution. You can see that for both of these, there's a higher incidence at younger ages, whereas for some of these other molecular features, the age incidence is higher later in life or kind of in midlife. Another interesting feature of these mutations is that it seems to kind of track with where the tumor is occurring in the brain. And the K27 mutation in histone H3 is the most striking example because the brainstem tumors, this is kind of exclusively occurring in the brainstem when the K27 mutation is present. So we have to always ask, are these mutations the cause of the cancer? Are they the consequence of the cancer? And that is what is still kind of being unraveled. And then, of course, if they are contributing to the cancer, we want to target them. So there have been careful studies of the K27 mutation by many different groups, but one group in particular, David Alice's group, who's like a epigenetics rock star, a cancer epigenetics rock star in New York City at the Rockefeller, has taken this mutation and has characterized kind of what it means in terms of some of the readers, writers, and erasers. And he's focused on a complex called the polycomb complex. And what he finds is that the polycomb complex is actually inhibited, and this is Western blotting for some of the marks associated with the polycomb complex. This is immunohistochemistry, and you can see that in the presence of the K27 mutation, there's less staining, which tracks with less of this reader complex. So this is telling us that the mutation actually translates to less activity of this epigenetic complex. So we don't want to inhibit that activity. It's already down as a consequence of the mutation. Instead, we want to figure out maybe ways to increase it. And more importantly, we want to figure out what it means that it's down in terms of, is it an oncogenic event or does it require an additional cooperative mutation? So that work is ongoing. So things like the DOT1L inhibitors, the bromodomain inhibitors that I mentioned relative to MLL-AF9 are still in early phase clinical trials. They are not FDA approved. I'm a translational researcher, so I'm a PhD working in a clinical department. I want to help my clinician colleagues figure out what they can use now. So what can they actually, what knowledge is out there that they can build trials around? What can they actually apply in the clinic? So in terms of what's available in the Epigenetics toolkit that's FDA approved, there are inhibitors, chemical inhibitors of proteins called HDACs, and we'll talk more about HDACs. HDACs are histone deacetylases. The inhibitors are diverse in their structure, and there are several of them that are FDA approved. Panabinostat was just FDA approved last week, so there's kind of continuing evaluation of different classes of these compounds, and there's, I guess to date, about 4 different HDAC inhibitors that are available and FDA approved. There's also inhibitors of histone methylation and demethylation. Some of these are repurposed drugs, and I'll talk about one of them. And then there's also inhibitors of, that are DNA hypomethylating agents. So they're acting on DNA marks, and Kristen can tell you more about those because she studies DNA methylation. I won't touch upon them as much, although we have looked at their mechanism of action in some settings. And again, we really want to see whether some of these drugs can be used in the settings that I described, because there haven't been very many new medicines approved for pediatric cancers in the past couple of decades. So the pace of epigenetic drug discovery is really accelerating. So there's many examples of already FDA-approved agents. Those are the hypomethylating agents, some of the HDACs. There's additional HDAC inhibitors being developed. There's inhibitors of some of the readers that are the bromodomain-containing proteins. I'll talk to you about a protein called KDM1A, also known as LSD1, which is actually a little further along now than it was when this figure was made. It's actually in clinical trials. So lots of new agents are coming forward in terms of trials and late-stage trials. So the HDAC inhibitors are a class of drugs that are kind of the most abundant in terms of being FDA approved, and basically what they do is they inhibit the activity of histone deacetylases. Histone deacetylases take marks off of histones. The mark that they take off is an acetyl mark, hence the name, histone deacetylases. And so they are a reader, writer, or eraser. What are they? Yay! Very good, Chris. I'm impressed. The non-scientist. All right, so they're an eraser. They are antagonizing histone acetyltransferases, which are writers. So that's the class of compounds that they're antagonizing. So ultimately, what they're doing is promoting hyperacetylation and promoting this kind of change in accessibility of the DNA for transcription factors to sit down and ultimately affecting gene expression. Well, there are 11 different HDAC family members. It's a superfamily. And in terms of the drugs that are available, they fall into very different chemical classes. So there are short-chain fatty acids, hydroxamic acid derivatives, benzamides, diverse chemical classes. Here are some of the names of the drugs. I'll be talking to you about vorinostat, which you can see is inhibiting all of the HDAC family members, so it's a pan-HDAC inhibitor, but there are pharmacological tools that can kind of hone in on one or two specific HDACs. So these chemicals can be used as a way to kind of unravel the biology of how these HDACs are contributing to disease. So for example, entinostat, which is a compound that we have worked quite a bit with, but I won't be talking about so much today, is a class 1 HDAC. It's only inhibiting HDACs 1 through 3. The pan-HDACs, like I said, vary quite a bit in terms of their efficacy, their potency. Structurally, they're different, and it can't be assumed that if you have two classes from this drug, they're going to act exactly the same, and that's kind of what led to the approval of panabinostat last week. It was effective in a multiple myeloma setting clinically, much more so than vorinostat. So you kind of have to take the drugs one by one and understand how they're working and what setting they're going to be most efficacious in. So this is just a kind of a closer look at the HDACs and the HDAC inhibitors. Here are the family members that the drugs are antagonizing. SAHA is the old name for vorinostat. LBH589 is the old name for panabinostat. These drugs always have zip code names when they're owned by the company, and then when they get further along, they get different, really long, hard to remember names. So, all right, so the histone deacetylase inhibitors are basically acting on acetyl marks, and this is kind of a close-in view of the histone tails that May mentioned, and you're getting a sense of all the modifications that occur on histone tails. So each of these lollipop-looking things is a modification. In the case of the HDACs, the modification that we're hitting is the acetyl mark, but you can see there's other colored marks too. There's phosphorylation, there's methylation, that's the ME mark, there's many marks that aren't even represented here. I actually have a slide that I took out that had like 25 different modifications that can occur on histones. Some of them have names like crotonylation, and it's not even known what it means to crotonylate a histone protein, but the mark has been described and it's out there, and it must be doing something, right? But this biological significance still has yet to be identified. So we have focused on the acetyl mark and the methyl mark, and as with all the marks, there's readers and writers, right? So The histone deacetylase inhibitors are antagonizing an eraser, and we got interested in whether there was interplay between some of the acetyl marks and some of the methyl marks, because if you'll notice, there's a couple of marks that get both, or a couple of residues, sorry, that get both. And on histone H3, lysine 9 is one example where it can be methylated or acetylated. Furthermore, if a residue is acetylated, it would influence whether it can be methylated or not. It's a question of reach, right? Because you've kind of got occupancy on a spot, so maybe a reader, writer, eraser can't recognize it. So there's definite interplay between these marks. So we were interested in LSD1, lysine-specific demethylase. So it's an eraser. It's taking methyl groups off. And it acts upon H3K4 and H3K9. And if LSD1 is too trippy of a name for you, you can refer to it as KDM1A, which is the gene name, and kind of highlights the fact that it's a lysine demethylase. The name lysine demethylase also reinforces an additional layer of complexity of these enzymes because they not only act on histones, but they act on non-histone proteins. And this is where we need some really good proteomics work to figure out what all the substrates are for these enzymes. Some of that has been done in the context of the HDACs, where mass spec-based proteomics approaches have been used to look at these non-histone proteins, but there's like a whole constellation of things that these enzymes are regulating independent of histones, and they're doing it directly by modifying lysine residues on lots and lots of different proteins. All right, but we're going to focus on histones. So LSD1 actually demethylates H3K4, but H3K4 can have a monomark, a dimethyl mark, or a trimethyl mark. So these methyl marks pile on top of each other. It's not just a question of erasing one at a specific time. There's different enzymes that might act on a trimethyl mark or the dimethyl mark. LSD1 can act on a dimethyl mark, so two methyl groups stuck on H3K4 or H3K9 at once, or if there's one methyl group, it cannot act on the tri. So yet another layer of specificity of these enzymes. So we were intrigued by reports that LSD1 and some of the HDAC family members were together in complexes. They were co-localizing with these large transcriptional complexes, and that there was evidence of some of the drugs that I mentioned that are HDAC inhibitors actually causing accumulation of H3K4 dimethylation, so the mark that LSD1 is acting upon. So that suggested to us that maybe a strategy for enhancing the efficacy of the HDAC inhibitors could be to also inhibit LSD1. One of the reasons we thought that this would be something of high translational relevance was because there was a drug already available to us that was able to inhibit LSD1. And so this is the less trippy name for LSD1, KDM1A. This is the ribbon structure of LSD1 relative to monoamine oxidase B. I don't know if there are any psychologists or psychiatrists in the audience. We were talking about this last night, but there are inhibitors of monoamine oxidase B that have been used for decades for treatment of depression. And one in particular, tranylcypromine, has been shown to also inhibit LSD1 or KDM1A. And it's because, as you can see, their structures are so similar. Both of these enzymes are amine oxidases, so they share structural features in terms of their protein structure. So we have in hand a FDA-approved drug that hits LSD1. It was used for treatment of depression, so it crosses the blood-brain barrier, so it can be used for brain tumor treatment. So we were extremely excited about that. And so we set out to just kind of survey brain tumor lines for expression of LSD1 to see if there was differential expression in normal versus transformed models. We started with a panel of cell lines. We used human brain tumor cell lines and compared expression, protein expression in this case, to normal human astrocytes that have been immortalized to be maintained in cell culture. We also looked at patient-derived glioblastoma stem cells. So this panel of GSC lines are all patient-derived cell lines from patients with brain tumors where they have stem-like properties. They've been cultured in sphere culture. And we compared LSD1 expression to normal neural progenitor cells that we purchased that were basically from non-cancer patients. So you can get a sense of LSD1 expression being higher in the majority of GSC samples and LSD1 expression being higher in the brain tumor cell lines relative to normal human astrocytes. And then we tried this strategy of combining the HDAC inhibitor. In this case, we used vorinostat, which was already FDA approved, was known to cross the blood-brain barrier, again, readily available in our toolbox, and combined it with tranylcypromine, the LSD1 inhibitor, which is also FDA approved, used to treat depression. What was striking was that alone, tranylcypromine really didn't do much. Vorinostat is known to kill brain tumor cells, but we were using it at concentrations that were quite sublethal. But together, you can get a sense of how much of an increase there is in apoptotic DNA fragmentation, which is what we're measuring here. If you do the same experiment in immortalized astrocytes, so these cells, the normal counterparts of the human brain tumor cells, there's not this increase. And we've done the same experiment in leukemia lines, where we compared human leukemia cell lines to peripheral blood mononuclear cells from healthy donors, and we see the same type of selectivity. We see higher expression of LSD1 in the tumor cells versus non-transformed counterparts. So we think the strategy is applicable to more than one tumor type. And this seems to be effective in these GSC models too, which is kind of exciting because these are thought to be the stem cells that contribute to the disease. Here we're looking at loss of viability. The combination of vorinostat and tranylcypromine doesn't do so much alone, but when you combine them together, there's a drop-off in viability. And we've kind of subjected all of this to the mathematical models to calculate synergy and find that there are synergistic interactions at many different doses. My background is in cell death research, so I was really interested in understanding how this is causing cell death. And so we used RNA-seq to identify all the genes that are changed by either the HDAC inhibitor alone, LSD1 knockdown in this case, because we really wanted to implicate LSD1 rather than being criticized for having nonspecific effects of an inhibitor, and then the combination. And what we found was that there were some surprises. So P53, which is a well-known oncogene, was downregulated. And this could be good, right? Downregulating an oncogene to the point where it almost disappears. And increased in expression really highly was a pro-apoptotic protein called HRK. And so we think that this is one of the ways in which cell death is being triggered. We've also carried this into an in vivo model where we implant brain tumor cells intracranially into mice. So these are human brain tumor cells growing in mice in the right place. They're in the brain. And what you can see is that the mice that are treated have a reduction in the size of their brain tumors. The mice that have the combination treatment live longer than mice that get the single treatment alone. And the changes that we found in RNA-seq are usable in mouse models. So if we look at P53 expression, it goes down with the combination, and then a superfamily member of the P53, superfamily P73 also goes down. So those are some potential biomarkers that we think could report out whether this combination is working. So we're using tranylcypromine, which is already FDA approved. Vorinostat's already FDA approved, so that combination could be translated into a trial if we just had the money, the right collaborators, all of those little details. So in the meanwhile, there's been a lot of excitement about LSD1, and there are 2 ongoing clinical trials that just started last year using different inhibitors of LSD1, which are structurally similar to tranylcypromine. This is one of them from a Spanish company called Horizon, and then GlaxoSmithKline has another inhibitor, which is also in clinical trials as of last year, which is kind of a big deal because LSD1 was only discovered in 2004. So from the discovery of the gene to now, 11 years later, there's already clinical trials targeting this epigenetic enzyme. So it gives you a sense of the kind of exciting pace of drug discovery and the capacity to really participate in the process of bringing some of these findings to patients. So, so we're actually talking to GSK now about using their compound together with HDAC inhibitors, and hopefully that will kind of move forward at MD Anderson. All right, so I often feel like I am disguising myself as an epigenetics because my real passion is oxidative stress. And the reason that I got interested in the HDAC inhibitors and even in LSD1 is because they have links to oxidative stress. And so the HDAC inhibitors as a class of agents generate oxidative stress as part of their action. And LSD1, the enzyme, it's an amine oxidase. It generates a small burst of oxidative stress as it functions. And so that's really the reason I got interested in it initially. So my background is in oxidative stress, and we take different approaches in terms of viewing oxidative stress and its therapeutic potential. One strategy is in cancer cells, you have higher levels of oxidative stress because of increased proliferative capacity, because of increased mitochondrial function, because of increased kind of expression of endogenous sources of oxidative stress or altered antioxidant functions. So if you raise ROS levels even further in the context of treatment, then you can push those cells towards cell death. And so we think about that a lot in terms of some of the targeted agents. We don't want to think about it if you're just using an agent whose primary mode of action is oxidative stress, because cells are very adaptable in terms of upregulating antioxidants or downregulating antioxidants. So we want to couple that oxidative stress with another primary target. And that's how we kind of got into the HDAC inhibitors. And we also study the proteasome inhibitors, which are another class of compounds that have clinically FDA-approved, clinically relevant medicines that are currently being used for cancer. So that's kind of one of our ideas about how to modulate oxidative stress. Oncogenes, though, kind of rely on oxidative stress. So there's a concept of oncogenic addiction out there that maybe some of you are familiar with. The idea being that a cancer cell is dependent upon an oncogene for survival. And if you cripple the oncogene, which a lot of cancer therapies are doing, then you kill the cancer cell. Well, we think that a part of that oncogene addiction is an addiction to higher levels of oxidative stress. So if you starve the cells of oxidative stress, and this is kind of more in a progression setting, then you're going to cripple the cancer cell. And so we've shown that with examples of where we look at genes that are turned on by oxidative stress. Some of the pathways that are turned on by oxidative stress are shown here. If you knock out a component of the genes that are regulated by oxidative stress, then you slow down cancer growth, you sensitize them to some of the oncogenic inhibitors. So that's kind of another broad strategy that we use. So you can turn the rheostat of oxidative stress high or low depending on the context. So that got us interested in how pharmacological modes could be used to modulate oxidative stress. And we started thinking about diet and how diet might actually be used together with cancer therapy to promote oxidative stress. Because in the context of cancer therapy, there's lots of currently used therapeutics that raise levels of oxidative stress. So can you push that kind of envelope a little further with foods that are being consumed that might promote oxidative stress to help those treatments be more efficacious? So our idea was that dietary oxidative stress combined with chemotherapy-induced oxidative stress might improve responses. So about 5 years ago, I launched a program called On to Life, and the ON stands for Optimizing Nutrition, and this was funded by the Gerber Foundation that makes Gerber baby food, because they were really interested in the question of diet and oxidative stress and outcomes in pediatric cancer patients. And the project that I wrote up for the Gerber Foundation was really looking at measuring caloric intake in pediatric cancer patients and tracking that with actual measures of oxidative stress kind of in real time. So I'll show you a little bit of data on that. But as we started kind of consenting patients for these studies and interacting with parents, interacting with families, we learned there was a big need for guidance and resources related to nutrition. And so we started doing these things to kind of fill some of those gaps. We also learned that we had to kind of figure out how to change behaviors related to eating and exercise, and that's kind of a gap. So I kind of got involved in all of these angles, and it's been an exciting ride. So in terms of the place where we started, looking at oxidative stress in the context of pediatric cancer patients that are being treated for leukemia or lymphoma, we basically began an observational study where during the course of their treatment. We basically used a 6-month treatment course. We captured them in terms of their diet and their levels of oxidative stress in peripheral blood mononuclear cells at several different time points, one of which was chemotherapy naive for about a week. Ideally, we would have wanted to get them before they got any chemotherapy to get a sense of their baseline levels, but this was really difficult. So we tracked their levels of oxidative stress over a 6-month course, and what we found was that in the majority of patients, there was an increase in reactive oxygen species during therapy, and this is consistent with what we thought. Chemotherapies raise levels of oxidative stress, and so this was gratifying to see it borne out in this population. What was interesting, though, was that the younger patients had this correlation between vitamin E, which many of you think of as an antioxidant, and high levels of one type of reactive oxygen species, superoxide. So that doesn't make sense, right? Why would vitamin E be tracking with high superoxide? Well, we're trying to understand that more, and keep in mind this is just from regular foods. We're not giving these kids any supplements, we're not giving them any pills, but the kids under 4 are consuming more cereals that are fortified with vitamin E. They're consuming more formulas that are fortified with vitamin E, so they are actually getting superphysiological levels of vitamin E. And vitamin E and vitamin C are 2 examples of antioxidants that at high doses can redox cycle and can actually generate reactive oxygen species. So although we haven't proved that mechanistically in this exact study, that's what we think is happening, and we want to prove that point. But what this means is that perhaps a vitamin E-rich diet could help patients while they're going through therapy. And this is where we started thinking about resources we could generate to kind of give guidance to patients, because we were getting those questions a lot. What can my child eat while they're being treated? They only have specific cravings. So we made an electronic cookbook, and that was quite an adventure because we had to first collect recipes that would be healthy. We hired a research dietitian, collected over 500 recipes, and this is freely available, so anybody can use it. Under the search tab, you can actually click on symptom-related recipes. If a patient is suffering from dry mouth or mouth sores, you can click on that. It'll give you a series of recipes that might have a smoother texture, might have a more soothing flavor. And in real time, if you don't like leeks, for example, you can click on the little garbage can and everything will self-adjust in terms of the caloric content, the kind of macro look at the ingredients. On the back end of this, so kind of viewable to the researchers, are all the micronutrients associated with these foods. So we can say we want to have a vitamin E-rich diet designed for a trial. I can sort all the high vitamin E containing recipes and say, okay, these are your options to try for the next couple of weeks, and kind of design a personalized diet based on what our outcomes are. So we kind of went through a long process of collection, testing, categorizing them. I showed you the recipe view. We have video demos on the website. We want to add kind of more practical tools, and more importantly, we want to incorporate this into counseling and obesity prevention initiatives. Why obesity prevention? Well, obesity prevention is really important in the setting of patients as well as survivors of pediatric cancers. In patients, there's emerging data showing that obese patients don't do as well with regard to their treatment as normal weight patients do. And in survivors, there's a number of late effects associated with pediatric cancer therapy. A lot of them are secondary cancers. A lot of them are metabolic diseases and cardiovascular diseases. And so in red, I've highlighted all the conditions that are seen in pediatric cancer survivors that are associated with obesity. So reducing obesity in this population has potential to improve quality of life. We did a survey study to get a sense of where our survivors stood in terms of their behaviors related to health. And so the majority of them did not meet kind of guidelines that were related to physical activity and fruit and vegetable consumption. 45% of them were overweight or obese. In general, the national trends are that survivors are on par with age-matched controls in the US for obesity rates, which, as we know, is not good. But these patients have more to lose because they already have a disposition to the conditions that I highlighted. We also learned from them that they were interested in changing their behaviors, but they wanted to use interventions that were computer-based or that they could pursue with their parents. That led us into video games. Now, you may be thinking, why would you use a video game to change behaviors? Well, we had these awesome collaborators that have studied obesity prevention in healthy kids and have used video games successfully in that population at Baylor, and they have basically designed video games for diabetes prevention, hence the name Escape from Diab. It's really a compelling storyline. They've built a movie around it. There's an evil King Edes, who lives in the kingdom of Diab, and the protagonists are trying to get out of Diab, and so they have to actually meet goals that are related to diet and exercise in order to get to different levels of the game. So it's not really just about playing the game, it's about incorporating changes in eating habits and exercise to get through the game. So we tested this game in our population, and what we found was that they were responsive to the concept of playing the game. We had to kind of think about the storyline a bit because some of the endings were met with one of the characters dies at the end of the game, and that can be controlled dependent upon the changes in behavior that the player exhibits, and some of the subgames were difficult. So we want to modify this game and actually test it in patients and survivors. We're also trying to capture patients kind of at a critical point when they might be at risk of becoming obese, and that choke point could be when they're on steroids as part of their chemotherapy. So we've been delivering nutrition counseling to kids that are on steroids with the long-term goal of preventing future problems. And so this study is just now complete where it was a randomized clinical trial. We had an intervention group and a control group. The intervention group met with the research dietitian on a monthly basis, got counseling, and actually had an exercise component to it as well. And so we've gotten some anecdotal data about improved behaviors, and the hard data is showing us that they did decrease the number of calories they were consuming, but it didn't affect their weight. So we're trying to understand what's going on there. The steroids are, as many of you well know, affecting kind of the way that nutrients are consumed. So there's probably more to it than simply changing your caloric intake that's going to help these patients. So what are the next steps for these projects? Well, we want to build in some of the exciting wearable technology into our games, and so that's kind of the suite of grants that I'm writing now, using the Apple iWatch and some of these functions that I think everyone's going to have. We also want to use the electronic medical record as an opportunity for capturing and monitoring health behaviors. Then finally, we think that some of these resources have applicability to cancer prevention. And so can we reduce risk of cancer down the road in obese children? Because we know that obesity is a risk factor for many cancers. Can we use things like the cookbook, some of the lessons we've learned in terms of counseling to actually change behaviors in obese children and their families? And so we did a study in a local pediatrics clinic that serves primarily Hispanic families and children. We had to deliver the entire intervention in Spanish. But it was a 10-week intervention where we did cooking exercises with the families, exercises with the kids, and basically put together a curriculum which we now want to carry forward in hopefully an electronic setting, because this was quite personnel intensive in terms of doing this on a weekly basis, having all the components to do the intervention. But we thought quite a bit about the cost of the foods that we were teaching them to make. Everything is posted on the website. There's nested recipes where they make a base recipe and they can make 3 daughter recipes down the course of the week. So we try to think as practically as possible. And then the physical activity that the kids did used minimal equipment, accounted for weight status, and we have a paper in press kind of describing the feasibility of that. So in summary, the On to Life program is trying to help current patients, survivors. We're trying to take some of the lessons we've learned into the general public to reduce cancer risk. And we're really trying to contribute to the spectrum of translational research, but it takes a village. And I've been fortunate to have lots of collaborators and lots of great people in my group to work with, and this is my village. I want to acknowledge all of them, as well as our funding sources. So thank you for the opportunity, and I'd be happy to take any questions.
We do have a few questions, a few minutes for questions before we go on to our final talk.
Very good. Thank you very much. I've been trying to create a cancer diet for my patients. It's not a pediatric population. It's actually 70% obese women with endometrial cancer. And a lot of the research that I've done centers around or focuses around the concept of ketosis and ketogenic diets. And I assume that you've looked into that. My best understanding of that is it is more of an anti-oxidative approach. In other words, being on a ketogenic diet is anti-oxidative or reduces oxidative stress. And it appears to be, at least in some animal models, associated with reduction in tumor volume.
Yes,
and so
brain tumors, yeah.
that would be in antithesis of this concept of prescribing a low-fat diet to your patients, and I wondered if you could comment on that.
Yeah, so I think there's a lot of work to be done in terms of figuring out which diet components are actually kind of doing the lion's share of the work in terms of cancer prevention. For the ketogenic diet, we've followed that literature carefully, and there's a lot of really exciting data, especially in brain tumor models, where it appears to be showing synergistic effects with brain tumor treatment. The problem with the ketogenic diet is compliance. It's really tough to stick to, and even more so with kids. So we, per se, are not going to use a ketogenic diet. We may try a modification of it. In terms of the theory behind why it might be working, I would subscribe that you have to test the levels of oxidative stress before you can definitively say in your population there's a change in oxidative stress. And that sort of biomarker incorporation is going to be really critical to this work moving forward, because in general, in the field of diet and any disease pathogenesis, there's been a lot of negative data showing that dietary changes or antioxidant consumption or supplement consumption in large clinical trials doesn't have an effect, but the appropriate biomarkers may not have been incorporated.
Question opens over to Cheryl. Anyone who wants to line up at that mic is also welcome to do so.
A very nice talk. I did have, in terms of the, DNA methylation inhibitors and the HDAC inhibitors, one point on that is, they do act indiscriminately. I mean, this has always been one of my biggest concerns, because there are cases we do want these marks put on there, like, potentially other proto-oncogenes. And I just wonder, do you know if there's been long-term follow-up studies with these children that have received such treatments to see if they are more prone to developing other cancers down the line? Because I think we really need to know that before we just kind of we need those long-term studies, and not just even like cancers, but are they at risk for other diseases? I mean, again, I do a lot of work with neuroscience, and we see a lot of changes when you give the HDAC inhibitors in terms of just learning and memory responses.
Cheryl, that's an extremely important point. And the short answer is no, those studies have not been done because the HDAC inhibitors haven't been used in kids long enough to figure out what their long-term effects are. But there are much more targeted therapies that have been used in kids, things like imatinib or Gleevec that targets the BCR-ABL oncogene. There's a subset of pediatric leukemias that display one version of the BCR-ABL oncogene. So that's a very selective inhibition of that oncogene. However, even that specific inhibitor hits other kinases. And one of the consequences of kids that get that inhibitor over time is that they're shorter in stature. And so, do you want a short kid or do you want a child that's alive is kind of the question that we're grappling with. Certainly you want to examine the impact on development and try to design better ways around it. But I think in the short term we have to recognize that many of these kids are not going to make it through their illness if they don't get some of these treatments. And yes, we certainly have to think about long-term effects, but the short-term goal is quite urgent for some of these subtypes of pediatric cancers.
I have to ask, what's up with the disembodied torso of the Pope?
Oh, I always get that question. So, grant funding is so hard, and we're always like trying to figure out ways to increase our chances. And so, no, so this is actually a restaurant in Houston where it's an Italian restaurant, and they have a room called the Pope Room. And the Pope Room has all kinds of Pope paraphernalia, and Pope John Paul is in the middle of this table in the Pope Room. They didn't have any pictures of the new Pope, which I thought was interesting. I don't know if that was a political statement or not, but yeah, that's the Pope Room at Italian place where we had our holiday dinner. Thanks for noticing.
Okay. I have a question about the antioxidant use. You referenced
sorry,
like
the what that I used?
antioxidant use in like children with like vitamin E and vitamin C. Whenever you're recommending, or like from studies that you've read, do you recommend like supplementation through like multivitamins, or do you just recommend it through like food sources?
So I'm not a clinician, so I don't
Right.
recommend anything. But what I tell clinicians is that they should not recommend supplementation because of the point that I kind of mentioned. Some of these antioxidants have redox cycling effects and can actually generate oxidative stress at certain dosing at certain doses. And so there's also a lot of data showing that specific antioxidants can interfere with drug action. So the general guidance that's given to most cancer patients is to get off of supplements, to get off of some of these natural products that have antioxidant properties, or at least inform their oncologist about exactly what they're taking so that they can look to the literature to see what's known about interactions.
Your tumors were p53 downregulated, as I recall from that slide. Can you remind us of the relationship between p53 and reactive oxygen?
Oh, yeah. Yeah, so we don't think that's relevant here, but I'd be happy to tell you about that. So wild-type p53 is probably acting as a pro-apoptotic protein in terms of translocating to mitochondria and promoting, so it's complicated. So p53 itself can have pro-apoptotic functions. p53 is a transcription factor, and it promotes the expression of lots of pro-cell death proteins, things like NOXA, PUMA, BID. These are all pro-apoptotic proteins that p53 promotes. So this result was weird to us because we did not expect p53 to go down in a setting of increased cell death. p53 should go up, right? If it's pro-apoptotic and it's contributing to cell death, why is it going down?
If it's wild-type p53.
Good point. So we tested that. So we looked at several different mutations of p53, overexpressed them in a background of null p53. The problem with doing this experiment is that anytime you overexpress p53, we saw cell death because p53 is pro-apoptotic. So we don't have a satisfactory answer. I can tell you that in cells that lack p53, so they're p53 null, they don't have the same response to the combination of drugs. So there's some element of sensitivity to p53 expression. And we actually think that the p53 disappearance is a nice thing about the drug because it actually works similarly on wild type versus wild type and mutant p53. So it would make mutant p53 go away as well. So it's kind of doing away with that oncogene. But we haven't figured out how to kind of explain the mechanistic role for p53, and for now, we see it so consistently and to such a great degree in terms of the decrease that we're thinking about it as a biomarker for the drugs working, because that's something that we see reproducibly. When we use the combination, p53 goes down, and it goes down to a significant measurable degree even in the in vivo material.
One last question. Go ahead.
Actually, it's kind of going off of the question about p53. It was actually suggested a while ago, I can't remember how many years ago, using a gene therapy or a viral therapy, using either like a p53 or a PKR upregulator that would be specifically targeted to these tumors. Mm-hmm. Given now what we just discussed about the epigenetic stuff, the readers, writers, and erasers, could that potentially pose a problem for the gene therapy approach, or
Great question.
what are your comments on
You need
that.
to be the one to attack that question. So I don't think that that's been adequately addressed. There are adenoviral P53 approaches being developed for cancer treatment. I don't think the epigenetic implications of those therapies have been looked at carefully, and I think it's certainly worthy of further study and better understanding. Good question, Austin.
All right, thanks very much, Joya.